Okay. Last week I couldn't upload it because a couple of you came up here. What is that? And I couldn't delete it. I could not delete it. I didn't know. I don't know how to use a Google phone. But a couple of you. One.
No.
Two. She was actually on the thumbnail. So I had to delete you. Yeah, you were on the video. Yeah, you and her. When you came with the bonnet, Professor, I have a question. And you were all in the video. Anyways, okay.
My bad.
So, it doesn't matter. So you didn't know.
I didn't know. I didn't even know that was a full recording.
It's okay. Now I don't even know how to delete this part now. Okay, all right. So we have to know where we are. When we're doing lab, we have to know what are we concerned with. So what is this lab all about? We talked last week that we cannot look at our enzyme in isolation. We have to know where exactly does it fall. You know, they say you cannot see the forest for the trees. So somebody cannot see the general picture because they look at something that is so minuscule. So our experiment is about the enzyme peroxidase. But it's not really about the enzyme peroxidase. It's about the properties. I don't want my voice all in the hallway. Can you, Christopher, please close the door? Yeah, no problem. Thank you so much, sir. Okay, so we're not doing the enzyme peroxidase for itself. We're looking at the properties of an enzyme. Does the enzyme have an optima? What's an optima? An enzyme optima. This is all in your exam. An optima is the proper setting for the most efficient production and function, for the most amount, quality and quantity for the enzyme. In order for the enzyme to do its job, it needs optimal pH, optimal temperature, optimal salinity, optimal concentration, okay, and so on. So we said that the story began with normal oxygen, O2, that became a free radical by gaining an extra electron, and it has turned from oxygen into
SOD.
nope, into super oxide anion because it has a negative charge. Then our body is equipped with built-in enzymes that counter this reactive. This is an ROS. The negative superoxide is a form of reactive oxidative species, and that causes oxidative stress. Oxidative stress. This oxidative stress needs to be neutralized, and that's where, as Sophie had said, SOD, superoxide dismutase, superoxide dismutase. You cannot bank on that review sheet. Okay, don't just say, okay, I'm not going to pay attention. Let me bank on the review sheet. I'll do it. I'll do fine on the exam. You got to pay attention. Superoxide dismutase is going to convert the superoxide minus into hydrogen peroxide, H2O2, which is a little better than this toxic reactive oxidative species. H2O2 hydrogen peroxide is not a reactive oxidative species, but it's not the best. It's not water. So we need to convert it to water, and that's where our enzyme, our very important enzyme, known as, what family does it belong to? It belongs to a family known as catalases. So we have different types of enzymes, but catalases are the ones who deal with reactive oxidative species. And the family is oxidase. But remember, because you have the same last name, it doesn't mean you are your own sibling. So you have to have your own name. That's peroxidase, because it deals with hydrogen peroxide. Peroxidase. 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 So now everybody following the story. So peroxidase is going to convert hydrogen peroxide. H2O2 is hydrogen peroxide, and peroxidase is going to convert it into water, H2O, plus normal oxygen. O2. Water, we use it for drinking. We use it for very important biological sustenance, while O2 is important for oxidative phosphorylation, oxidative phosphorylation, and aerobic respiration. Okay, so what is our experiment about? Our experiment is here. We have hydrogen peroxide. Do we have hydrogen peroxide? Yes, we do. Can you see hydrogen peroxide in your bench? Yes, it's right here. So hydrogen peroxide. We're gonna use hydrogen peroxide, and that is our reactant. That is our substrate. That is our substrate. That is our reactant. Reactant. Our substrate. That is our reactant. Then catalase, peroxidase, that is our enzyme. That is our enzyme. And water and oxygen are our product. So what happens now? What happens is we have this guaiacol. Can everybody see guaiacol? We already identified hydrogen peroxide. Now you have to identify your guaiacol. This guaiacol, what does it do? Guaiacol is colorless. Guaiacol. Guaiacol is colorless. What color is guaiacol? It's colorless, normally. But when it finds oxygen, it's going to become amber brown. Amber brown. Okay? So if I have a test tube, let's do this really quickly. If I have a test tube, and on my test tube, I put hydrogen peroxide. Let me put hydrogen peroxide on my test tube. If on my test tube I put hydrogen peroxide, can everybody see hydrogen peroxide?
Yeah.
So what did I put in my test tube now?
Hydrogen peroxide.
I mean, did I put product or reactant?
No.
From the formula, what did I put? Hydrogen peroxide. Is that a product or a reactant?
Reactant.
What is that? Yes, you. What is that? I just said it. It's right here. Substrate, reactant. So that is a reactant. Okay. Then, reactant. Then I'm going to add my enzyme. So before I add my enzyme, let me first add my marker, my guaiacol. Should my guaiacol, what color is guaiacol? What color is guaiacol? It's clear. So I did not lie to you. It's clear color. Now when will it change color? When will guaiacol change color? If there is oxygen. So now if I put guaiacol now, is it gonna change color? No. So let me put. You see, did not change color. Now, what's missing? I have reactant and I have product. What is missing? My hydrogen peroxidase. Where is my hydrogen peroxidase?
Is the hydrogen peroxidase the enzyme?
Yeah, hydrogen peroxidase is the catalase, the enzyme.
What does that say? Oxidase?
Oxidative phosphorylation. You asked me last week, aerobic respiration, oxidative phosphorylation. We write things down and then we forget what we wrote because we don't review it at home.
Yes, we do.
Yes, we do. I hope. Where does somebody have like a pen? Yeah, can I see that? Amazing. I used all of your pen caps. Okay, so now, so that's my enzyme.
Oh
Yeah. We fought for it. Okay, all right. So now I'm gonna put my enzyme. What do I expect?
Wait, Professor, it's already.
Because there's a little bit in there from before.
Okay.
Let me use a better one.
Okay.
You canceled my whole experiment.
West.
You canceled my whole experiment. Let's try again. Okay, guaiacol.
Right? It did not change color.
Guaiacol is colorless. And let me add hydrogen peroxide. Where is hydrogen? Right here. Hydrogen peroxide. You see? It did not change color. So now let me add So now let me add the enzyme. What's gonna happen? Just a tiny bit. Just a tiny sprinkle. Sprinkle, sprinkle, sprinkle. Now stir, 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 stir. So what's gonna happen? What does that mean? So on the exam, when I ask you when it changed color to amber, when it changed color to amber brown, there, amber brown. What's the name? Amber. What's the name of the singer? Amber something.
Amber Rose?
Amber Rose. Yeah. It changed to amber rose. I mean amber brown. Why did it change to amber brown? Yeah. Oh, that one you need to know that one. This one you need to know that one. When does it change to amber brown? When does it change to when is it clearless? So now that means we have oxygen. Because if we don't have oxygen, it's always gonna become colorless, clear. Excellent.
So when you add oxygen, it becomes amber.
We didn't add oxygen. The oxygen has already been made. Has already been made.
So if you ask us like,
You know that color.
I don't. If you ask us, why does it change to amber brown?
Because it detected a product. It detected there's oxygen. Okay, are we clear?
Yes.
Okay, now, so what is today's experiment about? So now today's experiment, what is the entire lab about? What are we testing? Yes?
Changing the pH.
Changing the pH. Yeah, that's what we're doing. But what is the lab about? What is the lab about? The lab is not about oxygen. The lab is not about hydrogen peroxide. The lab is not about catalyte itself. The lab is about enzyme properties. So now what I'm going to test, what we're going to test, we're going to test, what if I mess up with the temperature? If I increase the temperature, am I still going to have product? If I change the salinity, temperature, okay, if I change the pH, if I change salinity, if I change enzyme concentration, so the more enzyme I add, am I going to have more product, enzyme concentration? Or what about substrate concentration. So all of these factors, we're going to test them. We tested a couple last week. Today we're just going to look at the pH. That's why you have different bottles of different pHs. So what are we going to do? We're going to have different test tubes. Test tube number 1, number 2, number 3, number 4. All of them, they will have the same amount of reactant. They have the same amount of hydrogen peroxide, H2O2, 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 H2O2. They're gonna have the same temperature, so it's not gonna change. Room temperature, 25 degrees Celsius, 25 degrees Celsius, 25 degrees Celsius, 25 degrees Celsius. They're also gonna have the same what? They're gonna have the same enzyme. They're gonna have peroxidase, the same amount. Peroxidase, 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 peroxidase. Everything is the same except one thing. What's going to be different? The pH. So one is going to be pH 7, one is going to be pH 5, one is going to be pH 3, and one is going to be pH 9. I'm going to test all the same except the pH is going to be different. And what I'm going to do is, how are we going to be able to tell? How are we going to be able to tell which test tube had the most product? Well, obviously in lab you can say, well, test tube number 4 with pH 5 was really dark, okay? But on a journal article, on a scientific journal article, you cannot say, well, pH 5 was the most optimal pH for the enzyme because it was really dark. It was really dark. This one was really dark. And pH 5 was a little bit dark. It was a little bit dark. And pH 9 was kinda dark. We could not talk like that in a scientific paper.
If you want to say it's really, really dark, how would you say extremely?
It's extreme concentrated.
But we're not going to do that either.
Why not?
What?
We're not going to do that. Yes? Excellent. We're going to use the spectrophotometer. We're going to use the spectrophotometer and we're going to replace all of this with absorbance values. Absorbance absorbance, and transmittance. So, what does that mean? We're going to, and you need to know how the spectrophotometer works. I'm going to ask you about that on the exam. So you got to pay attention now and understand really well how it works, because it's very important. You're in biolab, you don't know how the spectrophotometer works. How does the spectrophotometer work? So let's begin step by step. Who is number one? We have a beam. What kind of beam?
White light.
Beam of white light. A beam of white light. You see the beam? Do you see the beam of white light? A beam of white light. The beam of white light is going to strike a... Enough talking. Just listen. You can discuss when you're doing the experiment. Filter. A filter. So number one, the beam of light. Number two, the filter or the prism. And what does the filter or the prism do? What do we say that the white light is? White light is a mixture of all colors. A mixture, excellent. It is a mixture of all colors. What is the filter? What is the prism gonna do?
It filters the light.
Excellent. So it's gonna divide it into red, Roy G. Biv, orange, white, yellow, green, blue, indigo, violet. Good. It's gonna, and now this order. Why isn't O at the top? Why is R at the top? Why is it ordered like that? Because this is, so why in the rainbow red at the top? Why not red in the middle? Yes?
That's the highest wavelength.
Excellent. That's the lowest wavelength. So the lowest wavelength, that's the lowest wavelength, and that is the highest wavelength. Then we're going to put our test tube, all of these test tubes, we're going to put them. We're going to put all the test tubes with their content. And then we're going to appoint a light. What is the light? What is the light that we designated for this experiment? What is the wavelength of light that was designated to be used in this experiment? Oh no. What is the wavelength? How many nanometers are we going to put on the spectrophotometer to project the light onward into our test sample? Oh no. Oh no. These help instructor guided quizzes are going to end, and we're going to have a written exam. If people are not going home and reviewing and studying and coming prepared. Do you guys want pre-lab quizzes?
No.
So prepare. Otherwise, I have to feedback and make these pre-lab quizzes if people are not coming, not even knowing what is the wavelength. So you have to study the experiment from before. You have to read the experiment from before knowing it. What are you coming to do? You can rely on me to explain to you. You have to study it and know it. And I'll be testing you on it. Yes?
So should we be buying one manual? Do you want us to read?
I posted it up for you last night. It's on the announcement. I told you don't have to buy it. I'll take photos. I took photos. I posted it up. All you got to do is assemble them and read them. You didn't check your email. Yeah, so bring it to class, read it. So from now on, bring it to class, read it, come prepared so we can have a discussion. So the wavelength is 470 nanometers. The 470 nanometers are going to strike our test sample, and our test sample is going to have behind it a screen. This screen is going to record for us how much light passed through, and that is known as transmittance, and how much light was impeded, how much light was not allowed to pass through, because there's a lot of particles. Yes?
So is 470 the standard amount of nanometers you're supposed to shoot at it, or is it just simply for this experiment?
Just simply for this experiment. Excellent. Simply for this experiment, we choose 470. But the law is, the amount of light that you will absorb at a particular wavelength, that's your fingerprint. No two substances will ever absorb the same exact wavelength, the same exact amount of light at the same wavelength. So at 470, different molecules will absorb different light. I don't want to take time from the experiment, but you have to understand that after we add all of our reagents, we're going to be putting them in the spectrophotometer, looking for both how much light passed through and how much light was absorbed. How much light passed through, how much light was absorbed. And our hypothesis, we have a null hypothesis. What's the null hypothesis? Christopher, what's the null hypothesis?
No, how do you pronounce
N-U-L-L. What's the null hypothesis? Yes, null hypothesis. The null hypothesis is the hypothesis that says there is no effect. Like, for example, you're testing the effect of temperature on enzyme properties. You say the null hypothesis would say it doesn't matter if the temperature is too low, if the temperature is so high, there will not be any change, statistically significant, detectable change in all of our variables. So if I have a test tube at temperature 10 degrees Celsius, 20, 30, 40, 50, 60, 70, 80, no matter what different, it does not gonna matter. If you change pH, so the null hypothesis here would be that the product and the absorbance and the transmittance will be the same no matter how the pH changes. If it's pH 3, 4, 5, 6, 7. That's the null hypothesis. It means that the thing we're testing is not significant. But the alternative hypothesis would say we have an optimal pH 5. The more we deviate downward or upward, the capacity of our enzyme function will drop, and that will be proven by the less absorbance. If there's less absorbance, that means there's less concentration. Less concentration, it means there's less product. Less product, it means the enzyme is not working properly. So we're going to find that in the lab. So now you have to understand, why are we using a spectrophotometer? How does the spectrophotometer work? And what are we testing? So without wasting time, let us now begin. You guys are going to divide into groups based on your spectrophotometer. Okay? You're going to, how many spectrophotometers are there? 1, 2, 3, 4, 5. So we need 5 groups, and divide equally. No group with like 20 and group with 3 people. This was what happened last lab. So make sure like you guys are dividing evenly, and each group, please appoint one member to come and pick 3 test tubes. Please come and pick 3 test tubes. Yeah. Take a Kim wipe and 3 test tubes for each group. So you guys are dividing into 2 groups. Yeah. So take a Kim wipe with 3.
We have 3 test tubes.
Yeah,
Are they clean?
Yeah, some that could be like user.
Yeah.
Yo, they're all clean. When would they come in the box?
Yeah, we're gonna just wash it later and throw it out. You can wear gloves from here. Make sure you put on gloves. Please put on gloves.
Evilish with his black glove. The glove is giving me.
Does everyone need to wear gloves?
Yeah, that's how it is. Yeah.
Professor, does everyone need gloves?
If you're gonna be touching the stuff, because we don't want to contaminate. We don't want to contaminate our cross-contaminate our samples.
I couldn't find where it has
I don't know.
478.
Yeah,
Okay, I'll show you.
because we printed out the sheet.
You printed it out. Did you read it?
Yeah, we're out the library.
Yeah,
That's
that's like
cool.
2 hours ago.
I'll show you. Okay, can you guys? Okay.
Assembly line.
You guys are lined up for your gloves so nicely.
Our gloves.
Okay, alrighty, every group has 3. Okay, now we were supposed to use this pipette. and we're supposed to use the pipette in order to.
Exactly.
I've been there.
Okay, all right. Okay, so what are we doing? We have 3 test tubes. Can you put them in the racks? Can you put your test tube in the rack?
But we don't have a rack.
No.
If you guys can drop them in the water container, the water sink, and later on, when you guys are rinsing yours, you can rinse them too, drop them back that way.
What are you trying to do?
Yeah, remove all the old test tubes. Remove them, please wash them. Remove them. Please wash, remove them.
No, they're not.
Run through your thing. I got it from this box.
Yeah, he just. Should we let us just get the rack?
The rack's right there. Okay? You guys good? You understand?
I think I was good.
Good. You want a lot of serves.
Finally.
100.
I took one. Calm down. This is
A
the
lot
last
of
one.
serves. Okay? Alright, next. What are you guys doing? You guys started your. Okay, alright. Let's now. So what do you have on our test tube? How many different fields do you have? No, there's a 9 one. Make sure that you take 4 test tubes. 4, not 3. I'm sorry. 4, not 3. So one more. Okay, so now take.
Because use the pipettes.
So the 4 of them are gonna have the same thing, except one thing that's different. pH. Okay. So put glycol the last one. So begin with labeling. Actually label all of them. The one that has pH 7, the one that has pH 3, pH 9, but everything else is gonna be the same. Everything is the same for the 3 except the pH.
I'm just gonna walk down to your house and like,
Okay, you guys are ready?
Yes, sir.
Now take, make sure that you take, put your pH now. Put your pH, just the pH. And label them. The one that you're gonna put pH 3, put pH 3. Label them. The front one is for calibration. But we gotta learn the different pH sets, right? We need to select the pH.
I swear guys, they tell me to roll it.
Dude, it's for pH 9.
Alright.
Don't trust me? What I do?
3, 7, 5.
Okay, I could put one more. This sketch is mad gay.
Do you guys label? So if you label,
But like, I'll hold it, but somebody else
you're
can
gonna
Good
put
enough.
about
pH
3
9.
drops of pH.
The pH is
We
like.
were supposed to use this, because remember we have to be very precise. A drop is not, so you're gonna use it, but that's gonna take forever. But use it. I want you to use it to learn how to use it. Okay? Get a pipette. Pipette. So use 0.5. Use 0.5 of the pH. Use about 0.5. So put it in there. Put it in there. Let me show you how.
Right, this was overnight shipping, like I had to.
And then get an amount.
I was like, I'm not, because my mom's so bald, my hair don't grow.
Spin it, spin, no, spin this, yeah. No, spin it. Yeah, there you go. Yeah. Yeah, that's what we're doing.
Well, I'm trying to do mine so well. Yes, you made it so well.
So now put 0.5. Get a pipette. Yeah, go look. Three more? You have more? Okay, yeah, good. Okay, so now, why do we need a pipette? Why not use a dropper? Be more precise. So, you do not use the same pH. Do not use the same pipette on the same pH. So take 5, like fill it up and use
Okay, all the way up there.
But it's gonna be like a lot, so that's why we need a dropper. This is for you to understand that.
That one's I did box it.
Not a whole.
So just have.
Yeah.
What are we doing? What are we doing, Professor?
Yeah.
So we're putting the pH inside the vial.
Hold on. What's going
We're
on?
putting the pH inside the vial.
Do not put different. Did you put different pipette in a different pH?
It's the same one. Just wait.
Just use it once, and for the rest use a dropper, because that's gonna take forever. You're gonna use the same pipetter. You're gonna use a different pipetter for a different pH. Yeah, this lab is supposed to be in 5 hours.
Where's the?
That's all. Okay, so do not use the same pipette in different pHs. Okay? You did that? So, assemble it. Figure it out. Assemble it. And then, yeah, and then spit it in the spinner. No.
We're putting the pH 9 liquid in the pH 9 vial.
8.5, put it into the respective.
Because we gotta have the same amount of one.
pH 9.
But it's
Yeah,
supposed to be
put
about-
your pH. pH first, yeah.
Okay.
On each one. But do not use the same one, right?
Yeah.
Yeah, but so that's what I'm telling you. Don't use a different pipette, now use a dropper. Yeah. For the rest, just use a
Use
dropper.
the pipette for one of them and then use the other for the rest.
But they always use the dropper. Just use the dropper.
He wanted us to try using the pipette.
Okay, we can try something like that.
Yes, so we can.
Now add your
Y'all really
hydrogen.
think like I'm like not hearing what you're saying.
Put like 3 drops.
Open the.
Put 3 drops of your, try to be precise. Put 3 drops. Yes. Yeah, wait.
Everyone's on my.
You're not gonna put this in the same one, right? Yes. So we're
What are
gonna
you
put-
doing,
Just don't pick up.
No, I think you're supposed to.
So you put
You're supposed to.
each pH in one test tube. That's pH 3, so you put it in test tube
Wait,
3.
it isn't there. Yeah.
Use 0.5. 0.5
Because it's clear.
like half. 0.5. Yeah. That's it. So use 0.5.
Yeah, okay.
Just put it in here.
Oh my God.
No, you're gonna cross contaminate it. So you're supposed to use one pipette for each pH. But yeah, this is just for you to show you how to.
It was literally like that. It was literally like that. Let's put it down.
So, stick it in there.
You open the.
Spin up. Spin up. Keep spinning. There you go. See? But don't drop a
She
lot.
don't put everything.
Just, yeah. So drop all of this and keep just like a 0.5. Keep going. Keep going.
Yeah, you
Yeah,
might.
that's
Okay,
fine.
so.
Yeah, no, drop it.
We already got pH 9. Yeah. This is 9. So pH 7. Did you put pH 7 yet?
No.
Which one's five?
7, 5, 3. I got 3. 7 is this one.
How many drops? 3?
Excellent. Yeah, and for the rest, you use a dropper. And it just to show you how to use it like that.
Okay, watch it.
So you did you put, so you put 3 drops
Count the
off.
3 drops.
The pH in each one, and you put it on the wrong side. Then the next is to put glycerol. Then guys put 3 drops of glycol. What is glycol?
He's trying to show you.
No, because I keep telling people. You don't want to. I'll burn them out
Upper brown
Period. Make him learn.
because so we tried
Mr. No,
formula was on. These are the formula.
Alright, let's the hydrogen. That's what we're supposed to do.
So only if there's
And now we have to start the experiment.
Oh, yeah. But they know we need heat. Okay.
So now look at the
So what are we supposed to add next? The hydrogen peroxide?
And like something. But it's not.
Alright, right now,
Where's the hydrogen peroxide? Hold it.
This has only pH and only glycol. So
you just said
Because we listening to Mr. Jonathan here.
Glycol and we have pH.
I didn't do anything, bro.
You're my Jonathan.
The enzyme enzyme. So if I add the enzyme, did you guys add enzyme to population?
But maybe pull up your government.
So if you add the enzyme,
Pull
you
up your
have
first and last step.
So you need the substrate. You know that? So add the substrate.
How
So now
does-
add 3 drops of hydrogen peroxide. Now you guys have everything. You have hydrogen peroxide. You have glycol. So now the only thing that's missing is
Oh,
peroxidase.
so we're supposed to add glycol hydrogen.
So they just equals 3 drops?
Enzyme. You guys want the enzyme?
Well, why are you making
How much you guys gonna pay? $10. Okay. You guys should pay. You guys pay. Where is the enzyme? Okay, that's the enzyme. Now we were supposed to weigh it so we can be precise.
Hold with caution.
We're not gonna go.
Make sure you're right when you put it on the.
Who gave me a pen cap?
Professor.
I need to scratch it out. Yeah, thank you so much. Is this the third one that you gave me? The same one. So we have
I
to
hope
be
y'all ever have
precise
to
with
do
it.
that shit. Man, flesh bro. I believe you got the one they will put under your tongue.
Yeah.
Like I had to have that
Even
one
more
right
little
here.
than that is enough. You see,
They
stop
had
now,
to hold
stop.
me down because I was
Look.
biting the nerve.
Stir.
So that's just like a little bit. Hydrogen peroxide. Oh my god, that is so strong. Wait.
You
You
want
remember
to
to
see my
have
full
hydrogen?
face? You guys want to see my full face? Okay, then go, keep going. Alright, so now you guys have everything. You have your pH, you have your enzyme. You have your...
This is the hydrogen peroxide. That's the enzyme. The enzyme is what makes it into the color. That makes it turn amber.
You have the reactant.
Because the glycol also changes it, but it takes a longer time. You too giddy, okay? You don't get it. Right now.
The hydrogen peroxide.
I tell you, it will.
I thought we already had hydrogen peroxide. Jonathan, watch it.
My hypothesis
This one I added a lot. This one I added a lot. Yeah. No, but you added a
I
lot.
think it's gonna be amber.
No, I added a lot.
I think it's
Wait, this is the lowest.
color.
And then you added like an even amount. So it is, so yeah, basically.
Guys, they used one of the Right? Girl, you better than stop. Alright, we ready for the color change. We're gonna turn into melanin and make it the black.
Okay, yes. Okay, so now what's the next step? Have one test tube, only water. One test tube, only water. So you can calibrate. So now what's the next step? You tell me what's the next step. To find out what's happening, so you need your, yeah, so go and make one, take one tube, label it as water for calibration. Yeah, just fill it with water. So it's ready? It has control one already? Labeled? Yeah, so put it. So, now make sure that the trend is.
We're about to start the actual
Jonathan,
Did you hear what the professor said?
he definitely did not. 'Cause he's busy flirting.
He said we
The
should be.
enzyme?
Wait,
Yeah, you guys have a cap?
We're gonna add it.
We have taken all of Jenna's.
I mean, it's right now. So we're gonna add a control vial with just water, but everything else is.
I'll give you your enzyme supply. Where do you guys have a paper? Okay, that's your enzyme. Okay, what do you guys need now? Enzyme.
we did the
We're using we're making a control first.
Okay, I'll give you the cap and put it on a Kim wipe. You guys have Kim wipe?
no, we did like a fresh one. Let's get a fresh one.
That is more than what you need. Yeah, that's fine. Do you guys need? You guys have already. Alright, so you need a blank. You need one for calibration. So get one test tube full of water.
Compared to all these.
Wait, how much water should I put?
You got the enzyme?
No, you're funny. I literally turned it on and it's like, wow.
So what happens?
You always put water before. And I'm gonna tell you guys how to calibrate. Who needs enzyme? Who needs enzyme?
Oh wait, no, we gotta put
Who needs enzyme? Have felt inside the thing. Wow.
So what's your name?
How can I get this out?
Christopher.
Yo, we've been saying it for the whole time.
I might have to call you
Now
Jonathan.
your hand is in enzyme.
That's what she said 20 minutes ago.
Yeah,
How can I get that out? Maybe I can use your glasses. No.
No, you can use this.
No, I cannot use that. That's a mom.
this one.
Guys, does somebody have a hairpin?
What?
So, how can I get this out? I'm in trouble now.
Oh,
Yeah, I'm in.
you got the cabinet there.
I got I'm in trouble.
I think I know.
Next class. Your professor is no longer here. Something happened. How can I get this out? You can use your nails?
I mean, French is very
I took French for Creole is it comes from French, like it's similar, like you got oh my god.
Get it out.
Jonathan.
Wait, does anybody have a scissor?
Oh my.
Fingernails, yeah, pinky.
Yes.
That don't look right.
She got it.
Oh! It fell.
You're right.
I don't have nails.
The more the pH lowers, the more the ink. So one ink
Good.
is a clear guy.
Excellent, thank you. I'm gonna go have nails.
It's not.
So,
Why?
yes, yes, we got it.
It's like oil. No, but it's still there. It's still here. Oh, damn, look at it.
Whoa. This one was my favorite color.
Okay, come on, girl.
What else?
Come here.
Yes.
I think the ice is
Why?
because
Yeah, because
I don't know what happens after that.
the alcohol pH is the one that.
So what are we doing this?
So when you put water, you have to set the how much it transmits speed. No, that's the wavelength. This is the transmit. How much should be transmitted? 100. It's water, so about 100 should be transmitted.
So this is the closest one you're from.
you're at 1 time.
Women
You're
can
way-
stand.
So make it 100.
Way off.
Yeah.
It looks
Very
exactly.
fucking nasty.
Okay. So.
Oh, yeah, that's
Okay,
the best
what if
part.
we hit it up?
Oh, there's.
No, that's not it.
Wait, that's
Then if you have water, how much?
Okay, I was actually right. I'm actually accurate right now. Yeah.
Jonathan, drink that and I give you $1 million right now.
Actually, I could drink it. I don't want to. I live it, but like,
That is not.
I don't think you got
These two are the same color and those two are the same color.
Oh! My brain just clicked. So you know how water is neutral. The more like the closer it's neutral, the more
So that's water?
no water is water.
And the
Like a water, shit. But like, if it's like high, yeah, or very low, it's not really that as dark.
Yeah, I feel like we're off topic right now.
It seems like what she got is like the more intense more neutral.
Because that's like really kind of like this. Enzyme on our
It
table.
really gives us any.
And then we added more and it got darker.
Don't drink it.
It's staying.
No, it'll stay.
Yeah.
One of the ingredients cancels out the. Right? You was talking about that. Which ingredient cancels out?
Water in there. How much do you think there is?
Glycol makes it so it's
Big what?
This does not smell like meat at all. It smells like brown
you don't like the sauce.
It's just like,
That's probably not good for you.
I think it's
I have the same way just eat.
that's so real that I'm still not.
Water doesn't have any molecules, it doesn't have any
Yeah, so that after I feel like that was. After you finish what you have
Look, when you open the lid, it changes. Because it's moving.
She's speaking the words like here.
Now I have Oh, I know someone.
Don't leave the minute classes. I don't know it's okay. Wait, they put all my effort into this. Christopher. I'm making some African tomorrow. Do you want to try some?
What is that?
It's like the African food, but I taste it and it's in the like we're gonna have it.
Oh, I didn't think of that.
What is that?
Yeah, kind of library.
You feel like you don't have to nice
You know, I don't think you can because like turn around. No, I'm gonna make for you. I'm gonna make some chicken. I still don't like the wings. And I'm gonna do
Okay, add water in there. Add like 1 milliliter of water. Now when you use Okay, listen up. Hold on. Everybody, stop, look, and listen. No. Listen up. I forgot to tell you something. You know you needed to add water to all the test tubes. Yeah. So that is a perfect way for you to use a pipetter. So go fill it up extremely with water and add to each of the 4 test tubes about a 1 milliliter of water. Add 1 milliliter of water to all the test tubes. And then we're going to do the calibration and take measurements after that. Yes?
Isn't that kind of standing to use pipettes for the other PH?
But we're not going to touch. So you just fill it up. Oh yeah, you mean for water? Yeah, you're definitely right. You need to have your own pipette that is only for water. Fill it up completely with water and then put like put 1 milliliter. Yeah, you have to put 1 milliliter. Put 1 milliliter of water on each one because that's what you're going to use to calibrate. Add 1 milliliter. So make sure you get it clean. All right, guys. Yes, what were you asking?
Jonathan said Christopher, the rest of them are good.
Was he still in? Yeah. Why is it off? No, that's a good guess. You need to make sure that turn it on. Let me turn it off.
Okay, hold on.
Back? No, turn it off.
He said just, how many, Professor? How many drops of water?
It's not the drops of water. It's about a whole milliliter of water.
So we have to add a little bit of water every.
Just add a milliliter of water.
Bro, do better.
Oh,
Jonathan?
wait, I just realized how this works. There's more water on my
Look at my
hands.
MacBook, bro. You're leaking.
Oh wow, we have only 15 minutes left. We need to. Okay. All right. So add the water. What we're supposed to be doing now,
Wait, hold on.
Okay, use that in 2 seconds.
because we gotta clean up.
If y'all don't be careful.
Guys, please. At 4:45, we have to clean up, put everything in the cart.
Jonathan.
Yeah, you can do it tonight.
Oh, so you don't have to you posted like some assignment, some announcement, you're like, oh, if you enter the quiz, you're gonna get reported to.
If you weren't in class, if you were not in class and you try to like from outside,
You see, you still consider that cheating?
yeah. If you're not in class and you take the quiz from outside, you're not here and it's this open at this interval and you're from outside trying to take it and you're not in class, that's cheating.
So what if we make an excuse absent?
That's different. That's excuse.
Are you supposed to like vote like that?
Guys, I'm not sure that everybody heard me. No leaving at 4:45. We have to clean up and then everybody can go. Okay? Yeah. It's new laws that we, yes.
The quiz, so it's fine if we do it on
Yes, you can. I think we can all do it together right now. I tell you, it's not a big problem. And don't forget your quiz. I made it up for you. Like you can do it tonight, yeah.
But I thought you said we can't do it because we're not in class.
I'm allowing you to do it. If you weren't in class and took it, that's a problem.
Okay, I gotta put some more in.
Okay, all right. So, what are we doing now?
Put the water. Put the pipettes and the stuff on. This one is not. It's not making any difference though.
So, what are we doing now? As a unit time.
Oh, that's how you use it?
Bro, you just got some rules. Pastor, if you put the water added, it's not changing.
That's my fault. That's my fault. I thought it was like a warning. You know, like
Okay, can I have your attention, please, everybody? Can I have your attention, please, for 2 minutes? So, after we run the spectrophotometer, what are we gonna do? We're gonna plot for each test tube 2 graphs. 1 graph for transmittance and 1 graph for absorbance. So, for example, let's say we're doing for pH 5. For pH 5, what's gonna happen is, as time goes on, 5 minutes, 10 minutes, 15 minutes, you gotta understand that. 25 minutes, 5 minutes, 10 minutes, 15, 20, 25. So what are we doing? We are taking measurements on 5-minute intervals with a spectrophotometer for each test tube. So for pH 5, we're gonna learn that as time goes on, the absorbance increases. The pH, the absorbance increases like that for pH 5. Then for pH 7, for example, it comes out like that. That's for pH 7. So what does that tell you? That tells you at pH 5, That tells you at pH 5, our enzyme gave us more product here, because more product here than at pH 7. So here we have proven that there is more substance to absorb light at 15 minutes when you are in test tube pH 5 than when you're in pH 7. Does that make sense?
Yes.
And then for the transmittance also, we see that we begin at 5 minutes with so much transmittance, and on pH 5, the more time goes, we transmit less. The more time goes, we transmit less. For pH 7, we're going to see something like that. We're going to see something like that. So what does that show you? That shows you as time goes on, we do transmit less. But when you compare, for example, at 15 minutes, when you are at pH 5, you're transmitting more or less light than pH 7, which shows you at pH 5, you have more substance, disallowing transmittance and disallowing the passage of light, impeding the passage of light than when compared to pH 7. And we have to do it for all the pHs. So that's the point. That's why we use the spectrophotometer, to have transmittance and to have absorbance readings and be able to make data that reflects for us that this is the most optimal pH. And that is done for everything. For temperature, we're going to plot the same thing. At temperature 25 degrees Celsius. At temperature 30 degrees Celsius. At temperature 40, and so on. So you have to understand that when it's most optimal, we're going to have the highest absorbance or transmittance. When the enzyme is at its most optimal pH or most optimal temperature or most optimal salinity or most optimal concentration, I'm going to have the highest what? The highest transmittance or highest absorbance. I'm going to have the highest absorbance because I'm going to have a lot of product. When I have a lot of absorbance, it means I have a lot of product absorbing the light. So when I'm at my optimal pH, when I am at my optimal temperature, I impede a lot of light, more than if I'm not at my optimal. Does that make sense?
Yes.
The more are you at your optimal environment with a pH, temperature, as an enzyme, you will produce more product and you will absorb more light and you will allow less transmittance. But when you're not at your optimal, you will not have a lot of product, you will not absorb a lot, and you will transmit a lot of light. That's the key point of this lab. Okay? Alright, so now, let's go ahead please and clean up. Can you please do me a favor? Divide your group. Some groups, please bring all of the reagents here. And if you have any garbage, that's the garbage can. And if you have test tubes, can you please wash them and bring them back here? If you have a pipetter, bring everything back here.
Professor,
Yes.
we have a test tube in the here.
Yeah, can you remove it, please? Please bring all of the pipettes. Bring all of the... Please bring all of the pipettes, all of the test tubes, and place the racks back and wash all of these test tubes. Yeah, and just bring them back.
Wait, are those buttons? Maybe. That's not a test tube. Oh, but it was there already. No, we added it.
Can you guys remove those? Yeah. Remove those, please. Thank you. Yeah. No,
Coleman.
Professor?
yeah, the test tubes, you can just put them out. Okay, everybody.
Did they get upset last time?
Yeah, just pull it out.
It's not coming out.
Yeah.
Oh my god.
That would be
Not
good.
literally, yeah,
Wow,
That
it
would
changed
be
color.
What does that mean?
Colloid. It turns into a colloid. So what is that?
What is that?
It's water and? Can you please guys put all the racks back? Take the rack, put it back.
Wait, is that water and oxygen? Or
What do you mean water and oxygen?
Okay.
wait, is that
If your table should be clear, all the racks should be back. All your test tubes, please wash your test tubes, plees, and bring the wash test tubes and put them here. They can use them tomorrow.
Great question.
Thank you so much. Yeah, if it's stained, just like wash it and put it out there.
Wait.
Thank you, you too. Thank you so much. Thank you guys. Bye bye. Do your quiz at home. It's due tonight. Did you guys hear that? Second of October. Okay, I'll fix that. It's due tonight. Yeah, it's due tonight. Yeah. Your quiz due tonight. October. Bye bye, Alice. All right, bye guys. Make sure your quiz is due tonight. Thank you. Okay, put the racks back. People are not. Okay, whose group is this? Oh no. Whose group is that?
That was different before we came.
I mean, it's okay. I mean, I want you guys to watch it too. They did that the same thing over there with all these other things.
Sorry, my bad. Our table's never mind.
Thank you, Abigail.
Wait, I have a question. What was that?
The what?
That you were telling us? You said something about it's called something.
Looks like a colloid. A colloid is, okay, you have to understand what is a solution, what is a suspension, what is a suspension, what is a solution, what is a colloid, and what is an emulsion. So a solution is when you have small little solutes that are dissolved in a solvent, like water, like sodium chloride or glucose in water. That's a solution. But when you have a red blood cells dissolved in plasma, which is like water, red blood cells are huge. So that's not a solution. Even though they are dissolved in something that's water that's big. It's like when you drop sugar into a swimming pool and when like somebody jump in it, an elephant jump in the swimming pool. So the sugar is too little, like 10 nanometers and less. That's like glucose. And that's a solution. But if you are so big, more than 100 nanometers, like a red blood cell, that's a suspension. So blood is a suspension. Yeah.
So that means the suspension will be thicker.
Yeah.
That would be a
Oh yeah, that's a suspension. And then you have a colloid. Colloid is when you have protein dissolved in water. So that looks like a colloid. It's always jello and white. That's like a colloid.
It's cytoplasm in a way. Cytoplasm, like that type of consistency, but not in that way.
Yeah, perfectly. The cytoplasm is colloid. Yeah, you got it.
Okay, and is it just only those two, or there's more?
The colloid and then the emulsion. Emulsion is like, oil vinegar for the dressing for the salad. Like oil vinegar for the you're like
Yeah.
oil vinegar for the salad for the dressing.
Yeah.
Every time I buy salad with a nice